So hello everyone again. Our today's speaker is Michael Levin, who is a distinguished professor in the biology department at Tufts University, and also serves as a director at the Allen's Discovery Center at Tufts and the Tufts Center for Regenerative and Developmental Biology. And his work advances ideas from developmental and statistical biology and uses a combination of molecular biophysics and computational modeling to understand how biological systems store and process information at multiple levels of organization to guide their function. And Michael joined us here to give a talk, which name is Intelligence Beyond the Brain, Morphogenesis as an example of the scaling and basal cognition. And the talk will last for about an hour, and it will follow a Q&A session and discussion, so please hold your question for until then. And now, Michael, the floor is yours.
Well, thank you so much. I really appreciate the opportunity to share some ideas with you. And I look forward to the discussion after the talk. So, and then after that, if anybody wants to get in touch or find any of the primary papers, everything is at these websites. So I want to start by thinking about this guy. Needs no introduction. That's obviously Alan Turing. He was very interested in artificial intelligence or intelligence in general, seeking to understand what minds really are and how we could create them. And, you know, we'd already do create them. We have children all the time, but this is the kind of thing that Dan Dennett might call competence without comprehension. And Turing wanted us to have comprehension about it, to be able to make cognitive systems rationally. What may be less obvious is that at one point he published this, which is a paper on morphogenesis or spontaneous pattern formation. And so that would be useful to understand chemical signaling in development and things like that. And so you might ask yourself, why would somebody that's interested in intelligence and computation and things like that, why would they be interested in morphogenesis? And I think that he saw a very deep truth, which is that these are actually fundamentally the same problem. And I'm going to talk about that today. And I think that if he had lived longer and really kind of worked on this this symmetry between these two problems, I think not only computer science but biology would be much further ahead. But anyway, let's see what we can do with it here. So one thing that sometimes people think about is that you have these kinds of collective behaviors. So let's say there's a termite colony or a beehive or something, and people will say, well, that may be a collective intelligence, maybe, and that would be in contrast to what people often think of as centralized intelligence. So you may think of yourself as a centralized intelligence, but the reality is, of course, that you are also a collection of parts. In fact, all intelligence is really collective intelligence because every cognitive system is made of some kind of parts. And so we're going to dig into this issue of scaling and so on. And so what you're seeing here, this is a single cell. This is a creature known as a lacrimaria. There's no brain, there's no nervous system, there are no stem cells. This is just one cell, and the whole thing is a single animal. It's handling all of its metabolic, physiological, behavioral, and morphological needs at the level of a single cell. And so this is the sort of thing that we are all made of. We are all made of single cells that used to be independent organisms. And so it really highlights developmental biology as perhaps the most amazing of the sciences in the following way. We all made this journey across the Cartesian cut. We all start life as this, basically a quiescent oocyte, a single cell that's a collection of chemicals, most of which aren't doing very much at the moment. And then, so someone could look at that and say, okay, this is what people call just physics, right? This is physics and chemistry, and that's it. And then eventually, you get to be something like this. You get to be this, or maybe something like this, but you become a centralized, very complex cognitive system that might make claims about, hey, I'm not a machine. I'm a true mind, whatever that is, and so on. But that journey is slow and gradual. So we all start life this way, and then slowly but surely, the process that goes from physics to mind is basically embryogenesis. And for that reason, we really need to dig in and understand what happens during this process, because there really is no magical stage where some lightning bolt comes down and there's this bright line and says, boom, now you're no longer physics, now you are a cognitive system. There's no such place. It's a gradual, slow process. So we need to understand that. So the main points that I'm going to get across, I hope, today are these. First of all, that biology uses a multiscale competency architecture, which is basically a nested set of problem solvers in various problem spaces that all have their own agendas, and that navigation policies and competencies in these spaces are really fundamental to understanding the emergence of mind and the functionality of mind. I'm going to argue that in a sense, goal-directedness is a really powerful invariant for recognizing, communicating, building, and relating to really unconventional agents, really novel kinds of things. And then I'll trot out a specific cognitive boundary model for the scaling of cognition. And about a third of the talk is going to be using a very specific example, understanding morphogenesis as the collective intelligence of cells navigating a particular kind of problem space, specifically anatomical morphospace. And I'll talk about electrical networks, which are the mechanisms by which that cognitive scaling takes place. And it's important because I'm going to say a lot of things that sound conceptual or philosophical, but the key thing to realize is that all of these lead to very practical empirical predictions, which we've been working on for over 20 years, and they've led to all kinds of new discoveries in terms of new capabilities, and what some of which are hitting biomedicine now in terms of biotech and things like that. So this is not just philosophy. These are really practical kinds of things to think about. And towards the end, we're going to think about this idea that the competency of life at all scales enables a kind of synthetic bioengineering that provides a massively large option space for new bodies and new minds that don't have standard evolutionary backstories. And that's going to have implications for a lot of things, including ethics. So we'll touch on that at the end. So let's start thinking about single cells, of which we are all made. Here are some cells. So this thing is a single cell. Okay, look at the let's just think about the spatial competencies. Look at the amazing order that was created by a single cell. This is not a misprint. This is a correct scale. This thing is huge, about 10 centimeters. This whole thing is a single cell. This is an alga called acetabularia. It's got some roots, it's got a stalk, it's got a cap, one nucleus. The whole thing is one cell. And so we start already understanding that single cells are not kind of some simplistic building block. They have some interesting morphological competencies. They also have behavioral competencies. So what you see here, let me just talk over this first. This is an experiment with a slime mold known as Physarum polycephalum. It's placed in the middle. That's the yellow portion. The white discs above and below are glass discs. They're completely inert. They have no food residue on them or anything like that. They're just glass. And the whole thing is sitting on a flat agar dish. And what happens is and this is reliable. This happens all the time. So it grows. This Physarum grows. And then what it will do is it will reliably choose to go to the heavier, to the larger mass. Okay. It always goes for the 3 over the 1. Unless you disturb it in various ways. And so what's happening here is that during the first few hours here, you can see it just kind of grows in every direction. And what it's doing is it's slowly tugging on the medium. It has it's a kind of biomechanical effect that's almost a kind of sonar where it tugs on the medium and it reads back very kind of minute differences in vibration induced by the strain angle that these glass discs are putting on the agar. So it can tell the difference at a distance of centimeters. It can tell the difference between 3 discs and 1, and then it kind of makes a decision and goes. After it's processed that information, boom, there it goes. So the interesting thing about this life form is that here the behavior and the morphology are intimately linked. So this creature, and again, this is just one cell. This whole thing is one cell. This creature's behavior is morphological change, right? This is an example of evolution slowly pivoting from problems in morphological space to behavioral space, and we'll get to that momentarily. So the next thing I want to do for a couple of minutes is to really kind of broaden our idea of what agents are and the continuity between different kinds of agents. And then we'll talk about some examples and then we'll move from there. So this is kind of the old classical picture of agents. This is a famous painting of Adam naming the animals in the Garden of Eden. And the reason this is important because this is really this kind of view pervades a lot of scientific research, which is this idea that what we're dealing with are discrete, separate natural kinds. You've got, people will do experiments and they will say, here is a rat, here is the human brain, and so on. And we really need to start breaking that down, because both on an evolutionary time scale and on a developmental time scale, these things very slowly morph into all kinds of other systems with all kinds of other properties. And it really becomes important to ask, when and how do these properties appear? And so let's just talk about some examples where things break down. This is a caterpillar. This is a kind of soft-bodied creature that needs to crawl around and chew plants. And what it has to turn into is a hard-bodied creature that has to fly and drink nectar. And because those take very different kinds of controllers to operate during this process of metamorphosis, when the caterpillar turns into a moth or butterfly, the brain is largely liquefied. Most of the connections are broken down, many cells die, and then a new brain with a new structure is rebuilt. One amazing thing is that, and you can read more about this here, is that actually memories are conserved. So memories persist, this incredible remodeling of the substrate. So that's pretty incredible. It has some implications for theories of memory that rely on fine-tuning synaptic connections and things like that. Also, you can think about what it is. I mean, it's one thing in philosophy 101 to think about what's it like to be a caterpillar or what's it like to be a butterfly. Let's go to the next level and ask ourselves, what's it like to be a caterpillar becoming a butterfly, right? What's it like to be a cognitive agent where, during the time that your brain is literally reformed to be a completely different kind of agent? So these kind of changes happen not only on evolutionary timescales, but also on the timescales of a living individual. Here's another example. These are planaria, so these are flatworms. They have a couple of interesting properties, which we'll talk about more shortly. One of them is that if you cut them into pieces, they regenerate. So each piece of the worm regenerates exactly what's missing, including the brain, and they do have a centralized brain. But the other thing is they're fairly smart. You can train them. And so and this was discovered in the 60s by McConnell, but we did it more recently using modern techniques. If you train a worm to recognize this kind of laser etched pattern as the place where they get their food, you then cut off their heads, you take the tail sits there doing nothing for about 8 or 9 days. It regrows a new brain. Once it regrows a new brain, now it could start to exhibit behavior. And then you find out that they actually remember the original information. And so what you can see is that information is moving from the brain during learning, presumably stored throughout the body in some fashion. We don't know how or where. Then when the new brain forms, that information is imprinted back onto the new brain so that you can see behavior. So the movement, so here you see an example where two things that are tightly linked are morphological memory, so the ability to restore the correct worm, with behavioral memory, which is things that you've learned during your lifetime, and the movement of that memory through tissue, right? The imprinting of the results of learning from various kinds of tissues onto a new brain. So the plasticity is kind of amazing. And it also exists in vertebrates. So here's an example. This is a tadpole. You'll see here's the brain, here are some nostrils, there's the mouth, there's the gut, here's the spinal cord and the tail. And what you'll see is that there's an eye that we induced on the tail, and we'll talk later about how that can happen. The eye induced on the tail, and what you'll see is we also engineered it so that there are no eyes in the primary head where they belong. So the only eye that this animal has is on its tail. And we just, in this case, it was just surgical. We just basically took the eye primordium and the early cells that are going to make an eye, we just stuck them on the tail. So there's the tail. And what we find out using this machine that can test and train these animals for different kinds of behaviors, what you find out is that these animals can see quite well. They can see and learn in behavioral visual behavioral trials. And so this eye, by the way, does not connect to the brain. It makes an optic nerve that comes out, starts looking around. Sometimes it hits the spinal cord, sometimes into the gut, but it does not go to the brain. And this brain, which evolved for millions of years to expect visual input from these two specific areas, now is able to say, there's this weird itchy patch of tissue on my tail. There seems to be some kind of signal coming from that through my spinal cord. No problem. I could recognize that as visual data and learn to behave accordingly. No multi-generational long-term adaptation needed. It happens in one set of offspring. So the plasticity here is amazing. And we can talk later about the implications of that for evolution, the fact that these kind of competencies really buffer the negative effects of mutation. There's a lot to be said about that. But that's, we're starting to start to get an idea for these different components. If you're a bunch of cells and you suddenly find yourselves next to muscle instead of next to the brain where you belong, you can still figure out how to make an eye. And if you don't find the optic tectum, you can connect the optic nerve somewhere else. So this notion of play the hand you're dealt, which is basically to have to figure out where you are, what you're doing, what your affordances are on the fly, and you can't really count on where you thought you were going to be, is important. So what biology makes use of is this multiscale competency architecture. Not only are we a set of nested dolls structurally, that part's kind of obvious. What's less obvious is that at each of these levels, all the way up through the swarm and beyond, each of these levels is a problem-solving competent agent. They solve problems in various spaces. It is not just whole animals that do it, but in fact, all of the components do it. And what do I mean by other spaces? Well, there's the familiar notion of behavior, which is clever things that organisms do by moving around in three-dimensional space. But there are other spaces like transcriptional space, so the space of possible gene expressions, anatomical morphospace, or the space of all the possible geometric layouts of a particular structure. This is what we're going to focus on for most of today. Or physiological space, which is the space of different physiological states. The thing is that when we're very good at many creatures are good at recognizing intelligence in this kind of space. So medium-sized objects moving at medium speeds in our visual field, we're good at learning the difference between rocks and cats and various other things, where we're good at estimating intelligence. We are terrible at estimating intelligence in other spaces because we don't have a training set. We normally don't perceive them. And imagine if you were born to have an immediate sense, the way that we have with vision, you had an immediate sense of your blood chemistry and all the clever things that your pancreas and your other organs were doing to maintain that despite various insults and so on. You'd have a much better idea of what competency in a physiological space looks like. So, from my perspective, when we assign and when somebody makes an IQ claim about a particular creature in some kind of space, and you have to specify the space, you're really taking an IQ test yourself, which means that you have to be smart enough to recognize intelligent behavior. You have to recognize the space in which that system works, recognize the kinds of goals it may have. That's very non-trivial. So my framework, what I'm working on, is a way to be able to recognize, create, and relate to, meaning in some cases meaning control, and really a wide range of diverse intelligence. So not just the familiar creatures like us and and apes and birds and maybe the octopus and something like that, but really let's get really wide. So all kinds of weird creatures, colonial organisms, swarms, also synthetic biology, engineered life forms that have never existed on Earth before. Also artificial intelligences, which may be just software, or they may be embodied robotics. Also eventually possibly exobiological agents, right? Where we're sort of looking at an N of one on Earth as the one phylogenetic course of life on Earth, and making conclusions about life and intelligence on that data set is kind of like testing your model on the same data that generated it. We really need these synthetic and engineering approaches to really get beyond what we see. And I'm not the first person to try for a model or a framework like that. So this is Wiener's and Rosenbluth's and Bigelow's kind of scale, where this kind of a cybernetic approach, which I like, which gets away from the idea of what are you made of and where did you come from, meaning evolved or designed or whatever, and really tries to make a rubric for different degrees of competencies or intelligences based on functional, observable, and in fact observer dependent kinds of classifications. So what I want is a framework that allows us to compare and control all of these kinds of things in the same framework, and it needs to may have new capabilities. So my central kind of claim is that all of these the kinds of things I'm saying today with respect to how to handle intelligence, cognition, and so on, is that it has to be empirically testable. These cannot be basically just philosophical kind of armchair preconceptions about what is and isn't intelligent or anything like that. These have to be experimental. And so so let's look at one example which is a morphogenesis as a collective intelligence of cells that has some very interesting competencies. So let's look at that. This is an example of an unconventional agent that we like to think about. So this here is a cross section through a human torso. So look at the amazing order. All these parts are the right size, the right shape, relative. They're sitting next to the correct tissues, angled in the correct way. Everything is where it's supposed to be in the vast majority of humans. And the question is, where did this order come from? Now, because we all start life like this, it's a collection of embryonic blastomeres, and these cells are going to build something like this reliably, very reliably. So, so one might ask in this process, how much intelligence is there? And I'll define that shortly. And where does this pattern even come from? You might be tempted to say DNA. But of course, we can read genomes now, and we know that what DNA specifies is protein sequences. So if you look at genomes, what you see are specifications for the micro level hardware that each cell gets to play with. But what you don't see directly is anything like the size, the shape, the type of symmetry, the number of eyes. Does it even have eyes? You don't see any of that in genomes. What you see is the microscopic hardware spec. So we really need to understand how this process happens. As workers in regenerative medicine, we'd like to know how to control it. So if we want to suggest to these cells that actually they should rebuild a piece that's missing, we'd like to know how to communicate with that collective intelligence. As engineers, we'd like to go further and say, well, this is nice, but what if we want the cells to build something completely different? What else can you get a set of cells with the same genetic hardware to do? What else can they do? And so the end game for that field, you might think of as the anatomical compiler. So someday in the future, we should be able to sit down in front of a computer and draw, not at the level of molecular pathways or anything like that, but literally the anatomy of the animal or plant that we want. So you could draw this three-headed flatworm like this, and if we knew what we were doing, the machine would compile that description down to a set of stimuli that would have to be given to the individual cells to get them to build exactly that. Okay, that would be proof that we've completely understood and cracked the way to communicate with the cellular collective. Now, why do we want this? Well, because in biomedicine, pretty much everything, with the exception of infectious disease, lands on this problem. So birth defects, regenerating after traumatic injury, reprogramming cancer, reversing aging, all of this relies on trying to understand this one basic question. How do collections of cells decide what it is that they're going to build? And if we knew that, we could solve all these problems. So it's a real important goal. We're very far away from this. We do not have anything. The community doesn't have anything like this yet. Now, why is that? I want to be clear about the difference between the software and the hardware. So here, just to give you a simple example, there are massive gaps in our understanding. So here's an axolotl larva, and baby axolotls have legs. And here's a tadpole of the frog, and tadpoles at this stage do not have legs. Now, in my group, we make something called a frogolotl. So a frogolotl is part frog, part axolotl cells early in the beginning. And now I could pose the following question. Well, we have both genomes. So we have the axolotl genome, we have the frog genome. Can anybody tell me whether frogolotls are going to have legs or not? And no one has the answer to that. We have absolutely no idea how to derive that kind of information from even given the full genetic information, because it isn't straightforward. It's a question really about the decision-making of a heterogeneous collective, and we do not have a good science of that yet. And so where we stand with all this is that we're very good at manipulating things like this. So pathways, we can rewire these pathways, and we can lots of single cell kinds of approaches, but we're really a very long way from the control of large scale structure and function. And today, molecular medicine is largely at the level of the hardware, meaning that everything we're excited about, CRISPR, pathway remodeling, all of these kinds of things are all down at the level of the hardware. And so from the control perspective, it looks like this. It's the way that we programmed computers in the 50s by literally interacting with the hardware. With biology, we are still basically there, where we've only just begun to scratch the surface and to really understand what other interfaces, right? Because the great thing about the modern computers is that they offer multiple high-level interfaces, meaning that on your laptop, when you switch from Microsoft Word to Photoshop, you don't get out your soldering iron and start rewiring. Why not? Because we have devices that have an interface that enables you to reprogram them by inputs, by stimuli, by experiences, not by hardware rewiring. Biology is still stuck here. So what are we missing? What's the big thing that we really need to understand besides the hardware? Well, this has been said many times before. William James had an interesting definition of intelligence, which I mostly use, which is it's a degree of the ability to reach the same goal by different means. It's the ability to harness your micro states and your various actions to reach a particular macro state, and to do so in different ways depending on local context, and to have different competency in doing that. So let's just think about I want to introduce this idea of a morphospace. So morphospace is basically it's a kind of virtual, multidimensional space that contains the variables needed to set a particular anatomical structure. So this is, for example, Raup's work where he drew a morphospace of every possible mollusk shell. So there are 3 parameters. There's some equation that I can't recall, where these 3 parameters will generate every type of snail and other types of shells you've ever seen, and all the ones that are possible and impossible. And that exist and don't exist in the real world. And here's that morphospace, and he sort of colored out which ones are the real ones and which ones haven't, don't really exist. But this is the space of all possible ones. So you can imagine that as you are an embryo trying to form one particular kind of snail shell, what your goal is to really navigate this space. You're trying to get from wherever you are to the correct region of that space for your species. And if you're a regenerative organism and you've been damaged, maybe you have now been sort of pushed into the wrong region. Now you got to find your way back. Interestingly, Darcy Thompson in the 40s already had this notion of morphospaces and deformations they're in. He showed these kind of grids of animals that when you deform the grid in a mathematical way, you get other species that also exist. Of course, he didn't have a mechanism for it. We're getting there now. So let's think about what competencies do living things have in navigating this morphospace. So here's one example. Like I said, all of us start out like this as this kind of shape, and we're going to get to this kind of shape, and it's generally extremely reliable. But not only is it extremely reliable, meaning that the system knows how to navigate from here to there, it actually has some really cool competencies. So for example, in mammalian embryos and many other kinds, if you cut the embryo in half, so here, what you don't end up with is 2 half bodies. What you end up with is 2 perfectly normal monozygotic twins. So each half knows the other half is missing, can recreate what's necessary. And so you've got this idea that from different starting states, the system can get to where it's going, the goal state, via different kinds of paths, and including getting around certain local minima, local maxima, things like that. And let's look at some examples of that. So this is regeneration. So this salamander, this is an axolotl, Mexican salamander. These guys regenerate their limbs, their eyes, their jaws, portions of the head and of the brain and the heart, in fact, and various other structures. And what happens is that if you cut anywhere here along the limb, it will regrow exactly what's missing, no more, no less. And then the most amazing thing is that it stops exactly when it's done. Now, when does it stop? It stops when the structure is that of a correct salamander arm. So this is a kind of anatomical homeostasis. It navigates morphospace. It can tell that this is not the right region. It will sort of navigate where it's going, and when it reaches the right region, it stops. Now, I should say that, and that's true no matter where you cut it, right? Up here at the distal end or back here at the proximal end, it makes exactly what's needed. So now I should say that regeneration is not just for so-called weird lower organisms. The human liver is highly regenerative. Deer regenerate massive amounts of bone and vasculature and innervation when they make their antlers, and even human children can regenerate their fingertips up to a certain age. So I've shown you what happens when you're deviated from the right position of morphospace, but it actually gets even much more impressive than that. This is one of my all-time favorite examples. This is a cross-section through a kidney tubule of a newt. And what you see is that there's usually about 8 or 9 cells that work together to make this lumen. Okay, and so this is a cross-section, so you can think of it going in the plane of the screen, and so that's what they do. Now, one thing you can do is you can create polyploid newts that have additional copies of the genomes, and when you do that, well, the first amazing thing is when you do that, you still get newts. You still get normal newts. It doesn't matter if you have 3, 4, 5, 6 times the normal genetic material, still get newts. That's already kind of cool. The other thing is that when you do that, the cells get bigger. When the cells get bigger, what you can see is that fewer of them are now working together to make exactly the same size lumen. So that's amazing thing number 2, that cell size is actually scaled proportionally towards the right large scale anatomical structure. The most amazing thing happens when you make cells truly gigantic. And when you do that, just one single cell will bend around itself, leaving a hole in the middle, making the same lumen. So what you have here, think about this. What you have here is, in this case, it's cell-to-cell communication. That's how they make these. That's how they make these tubulogenesis here. In this case, it's a completely different molecular mechanism. It's some kind of cytoskeletal bending by which the cell wraps itself around an empty space. So the system is able to call up different underlying molecular mechanisms in service of a large-scale anatomical goal. It's a nice example of top-down control, possibly top-down causation, but also it's a great example of this play the hand you're dealt concept, where these cells come into the world, they're making something. They cannot rely on not being cut in half along the way. They cannot rely on how much genetic material they have. They cannot rely on how big the cells are. All of those things are completely up for grabs. The system can't depend on any of that. It has to have the competency to make a proper salamander with all of those things, basically having to figure that out on the fly and using whatever other mechanisms it has to get where it's going. And we discussed that in a couple of these papers. So kind of a simple example of this idea of a morphogenetic goal we found a few years ago, where if you take, so again, this is a tadpole, right? Here in the nostrils, the mouth, the eyes. These tadpoles have to become frogs. And in order to become frogs, they have to rearrange their face. So the jaws have to move forward, the eyes have to come forward, the nostrils move, everything moves. And it was thought that somehow what the genome does is encode a hardwired set of movements, right? Every organ just moves in the right direction, the right amount, and then you go from a standard tadpole to a standard frog. What we did was, so we wanted to test that. And so the baseline assumption is that the cognitive level of that process, the baseline is that it's extremely low, meaning it's hardwired. It just does exactly the same thing. So we tested that hypothesis and we asked, well, maybe it's more intelligent than that. Maybe it has some competency to adapt itself. And what we did was we created these so-called Picasso tadpoles where everything is in the wrong place. The eye might be on the side of the head, the jaws are up here, everything is kind of moved around. And what we found is that these tadpoles still give rise to very normal looking frogs, because all of these organs will sort out relative to each other. They will move in novel, unconventional paths. Sometimes, in fact, they go too far and they have to double back, and until they get to a normal frog face. And so no, it's not a hardwired process. What the genetics somehow gives us is a machine that's able to do an error minimization scheme. It's not just the same action every time. It basically will move around until it reduces that error in morphospace to as low as it can. So we started thinking about extending this here is the standard kind of developmental biology paradigm, really focused on emergence. The idea is that, yeah, there's some genes that turn each other on and off. They make some proteins. And then there's this kind of parallel physics where things interact according to local rules, and simple rules give rise to very complex emergent outcomes. And there's your organism. Now, all of this is true. This does happen, and there is emergence. But as I just showed you numerous examples, the morphogenesis is not a feed-forward kind of a dumb process. It actually has feedback both at the level of physics and genetics, that when you are deviated from your particular target morphology, and that could be injury, it could be teratogens, it could be mutations, whatever. When you're deviated from that target morphology, these loops kick in that try to reduce that delta. Okay. Now, the one thing that's kind of expected here is that, well, biologists are very comfortable with feedback loops. Everybody knows there are feedback loops all over the place. But there are two interesting things about this that are novel. One is that you might have a feedback loop across something like pH, which is a simple scalar, and you're just trying to keep a range. We kind of understand how that might work. But here, the set point of this homeostatic process is a very complex, some sort of coarse-grained descriptor of what the anatomy should be. So now that becomes much more mysterious. How does a collection of tissue store a set point like that, a complex set point like that? But what it promises is that, well, if this is true, and if this really is a homeostatic circuit of the kind that explicitly encodes a representation of the correct goal state somewhere, then if we were able to find, learn to decode and learn to rewrite that pattern, we could make changes to the system instead of making changes down here, which is incredibly difficult. This is what's going to limit all of the advances in fields like CRISPR technology, genomic editing, all of that is going to hit a real wall after some low-hanging fruit, because reversing this is basically intractable. If you want to make large-scale changes up here, if you wanted to make a threefold symmetry axolotl instead of a bilateral symmetry, which genes would you want to change? We have no idea how to reverse this inverse problem is basically insolvable. Well, what we could do is change the set point and let the cells do what they do best, which is build to that set point. And this is why our thermostats are so great, because once you know how to change the set point, it doesn't matter how the rest of the system works. You don't need to rewire it. You don't need those micro level details to use it. So this model makes a very strong prediction. It makes a prediction that we ought to be able to find that explicit homeostatic set point, rewrite it, and let the cells build. And I'm going to show you exactly that. And in that, if that's true, it becomes an example of basal cognition. It becomes an example of a simple system that can store and represent a goal state and then work in morphospace to achieve that goal, despite various deformations and things along the way. So now, so how I started thinking about this was, well, how would tissue store a complex pattern, a complex anatomical pattern as a set point? Well, we have a good example of tissue that stores geometric patterns, and that's our brain. So when thinking about this, so now I'm going to show you how bioelectricity is used as an informational medium for these agents in their space. So in the brain, the hardware looks something like this. You have a collection of neurons that have ion channels in their plasma membrane. They set a voltage gradient. They then might communicate that voltage gradient to their neighbors according to across chemical and electrical synapses, for example, these gap junctions. And that network has some interesting behavior. So that's the software, that's the hardware. Here's what you're seeing. This group did an amazing video of a zebrafish brain. This is the electrical signaling in that zebrafish brain. And the commitment of neuroscience today is that if we were able to decode all that activity, if we understood the encoding, we would be able to extract from it the content of this creature's mind. We would know, what is it thinking about, what are the memories, what are the the plans, what does it want to do, and for example in the case of a maze navigation or something. So this is neural decoding, the idea that we should be able to decode and that all of the cognitive potential of the system lives in this electrophysiology, right? That's basically neuroscience. Well, it turns out that every cell in your body does this. So this is an extremely ancient system. All of the cells in your body have ion channels. Most of them have gap junctions with their neighbors. And evolution figured out that electrical networks were a great way to store information and use it for learning and other things long before brains came on the scene, basically around the time of bacterial biofilms were already doing some of this. And so we might ask, could we have a neuroscience outside the brain, basically use the same ideas of neural decoding and memory inception and so on in electrical tissues that are not the brain? And so we've started to do that, and that's what I'm going to show you today. In fact, one of the most interesting things is that while we have separate departments of developmental biology and neuroscience, we have separate journals, we have separate funding agencies, and we sort of treat them as separate things. The interesting thing here is that the techniques can't tell the difference. So this is a typical kind of architecture of brain and the idea that experience and neurotransmitters and so on modulate the behavior, and then what the brains do is control muscles to move you in three-dimensional space. That whole system maps quite isomorphically onto the same thing in the rest of your body if you just relax the assumption that these are neurons. So that's the symmetry. You just sort of ask the exact same questions about other cells, and you can take everything that's done in neuroscience, including all the perceptual control theory and other kinds of kind of conceptual issues, optogenetics, neurotransmitter molecules. Everything that neuroscience does is applicable outside the brain. The techniques do not distinguish. So that tells you something interesting about how firm are these boundaries that we've established. So so here the bioelectric networks outside the brain are literally the cognitive glue that holds these cells together and allows them to navigate a very different space than individual cells would ever navigate. And instead of controlling muscles to move you through three-dimensional space, what they do is move the configuration of your body through morphospace. Okay, same. Otherwise, everything's the same. So we developed some tools. We developed the first molecular tools to start to track and manipulate this. So these are voltage sensitive dyes. This is not a model. This is a real embryo. What you're seeing here is a frog embryo, and that now fluoresces in a way that reports the voltage of every cell. So you can literally see all of the conversations that these cells are having with each other. We do a lot of computational modeling here to try and understand how these voltage gradients relate to the different channels and pumps. I want to show you what some of the data look like. So this is a voltage sensitive dye. This is a grayscale instead of color, but it doesn't matter, same thing. This is an early frog embryo putting its face together. And this is one frame out of that video. And what you see is that long before the genes actually come on to pattern the face, it already has a bioelectrical pre-pattern of where everything is going to go. So here's where the eye goes, here's where the mouth goes, here are the placodes out here. Everything is already laid out. The reason I'm showing you this one is because this is like the easiest to decode. We call it the electric face because you literally can see the beginnings of the face here. Some of the other patterns are much harder to decode. They're just not obvious. But this one's pretty obvious. And so the thing is that whether when we start manipulating these patterns, so let's say I use optogenetics to wipe out this region of depolarization here and introduce it somewhere else, the gene expression downstream will change and then the anatomy will change. And so we can show functionally that this pattern is a required aspect of developmental control, right? It's a natural pattern. It's a natural part of the answer of how do cells know how to make a frog face. Now that's a natural pattern. Here's a pathological pattern. So this is an oncogene injected into this embryo. They will eventually make a tumor. The tumor will eventually start to metastasize. But long before any of that, you can detect that abnormal cell, that defection from the bioelectric grid. You can already see that oncogene has now triggered a depolarization where the gap junctions will close and the cells will just disconnect from each other. So you can see what these things look like. Now, more important than characterization are the tools for functional experiments. So basically, just like in the nervous system, there are literally two main things we can do. One is that with any tissue, we can go in and molecularly open and close the various gap junctions. So this controls which cell talks to which other cell. So that means you're controlling the topology of the network. And by the way, none of this there are no fields, there are no waves, no vibration, no magnetics, nothing like that. There's all molecular physiology. So we're using molecular techniques to open and close these gap junctions. And we can do the same thing with the ion channels to directly set electrical states. So we can use optogenetics, we can use drugs, we can mutate some channels. All of these ion channels and gap junctions are the interface that these cells are exposing to each other. Normally, who uses them normally? Well, other cells do, evolution does, certain microbiota does, and parasites and things like that. And now we as engineers are able to also use this interface. And what happens when you use this interface? Well, I told you that the eye was the position of the eye was determined in the head by a particular electrical state in that electric face. So we said, well, what if we move that somewhere else? And so we took some ion channel RNA that we knew would establish that type of bioelectric state somewhere else. We injected into cells that are going to be gut. And sure enough, these gut cells, when they get that electrical pattern, they will now begin to make eyes. Now, two things out of your developmental biology textbook are immediately shown to be wrong here. One is this idea that only this tissue up here, this anterior neuroectoderm is competent to become eye, that no other tissue can become eye. So that's wrong. That's just because the PAX6 gene is only able to work up here, but the bioelectrics can reset it anywhere. And one of the important things about this is that look at the way we are communicating to this collective. We are not specifying all of the information that's needed to make an eye. That's a lot of information. These eyes have lots of different cell types in very particular organizations. All we're doing is giving them a very simple bioelectrical signal. It's basically it's like a subroutine call. It's a modular trigger that says to the cells, build the eye here. The cells are doing all the heavy lifting. We provide very little information. That's how you know this is not a micromanaged event. This is altering the decision making of these cells. It's not us micromanaging which cell is going to become which cell type and all of that. And so it's very modular. In fact, and these eyes they can have all the right tissue layers, so lens, optic nerve, retina, all that stuff. There's another amazing component to this, which is that, okay, here, this is an ectopic lens sitting out in the tail of a tadpole somewhere. And the blue cells are the ones that we've modified with our ion channel that causes them to make a lens. So here they are making a lens. But these cells can tell that there's not enough of them to make a proper lens. So what do? They recruit their neighbors. These brown cells up here were never modified by us. They recruit them to make this very nice round lens exactly how it's supposed to be. So there are two levels of instructions here. It's us instructing these cells that you guys have to make a lens, but part of all the stuff that's downstream of that high level subroutine call is, and by the way, recruit whatever you need to make sure the lens is the correct size. We're not going to micromanage that for you. We don't tell you how big a lens should be. You already know how big a lens should be. And so what they do is then the secondary instructions, they instruct these other cells that they're going to be part of this project now. So what can we make? We can make oocysts. These are kind of inner ear balance organs. So in that same way, by misexpressing ion channels, you can make ectopic oocysts. You can make ectopic beating hearts. You can make extra forebrains. So you wonder like whether this animal is got this massive forebrain, whether he's any smarter than this one. We can make ectopic limbs. So here's our six-legged frog and so on. And we can even make fins. Now you might realize at this point that wait, tadpoles aren't supposed to have fins. Zebrafish have fins. And that's an interesting point we'll get to momentarily. So by controlling the pattern of bioelectric communication that tells all of these tissues what they should be, you get to make some very coherent large scale changes. In fact, we're using this in biomedicine now because frogs normally do not regenerate their legs. So you amputate the leg at this stage, 45 days later, nothing happens. We came up with a particular cocktail that opens specific ion flows. And what you see within just a few days, immediately you see this, you see pro-regenerative genes come on like MSX1. It right away starts growing the leg. So you've got your toe, you've got the toenail, you've got some toes here, and eventually you've got a very respectable leg that's touch sensitive and motile. And so now we're of course trying it in mammals because ultimately we'd love to have organ regeneration for human patients. At this point, I have to do a disclosure. So Morphoceuticals is a company that Dave Kaplan and I founded to do this. So Dave's lab makes these bioreactors that fit onto an amputation site, and then our ion channel drugs are inside of that, telling the site to make organs. So this is hopefully on its way to biomedicine. I'm going to switch now to a different organism and show you. Really, I want to literally show you an example of memory of storage in this collective intelligence. So these are the Planaria. I told you before that you can cut them into pieces. A couple of really amazing things about them. One is that they're highly regenerative. Number two, they don't age. So there's no such thing as an old Planarian. They basically regenerate. Anything that senesces, dies, they just regenerate it. So they are immortal. The worms that I have in my lab are in kind of direct physical continuity with worms that were here hundreds of millions of years ago. And so one might ask, how is it that every piece knows exactly what to rebuild to make a new worm, a perfect little worm? For example, how do they know they're supposed to have one head? Okay, where does that come from? I want to show you this example that we developed, which is, okay, here's a one-headed animal, one head, one tail. If you look at where the anterior genes are expressed, they're in the head, not the tail. And if you cut it 100% of the time with very high fidelity, that middle fragment is going to give you a one-headed regenerate. Now, here's an example. Again, one head, one tail. genes in the right, gene expression in the right position, except that when I cut this guy, I get a two-headed worm. Now, this is not Photoshop. These are real actual two-headed worms. Why is that? I just told you that this was a really reliable process. Well, it happens because what we did was we figured out that there was an electric circuit that enabled this, this fragment to know how many heads it's supposed to have. In fact, to remember how many heads it's supposed to have. And what you can do is, when you look at the bioelectrical pattern of that fragment, here's the bioelectrical pattern, and it says one head only. And so we can manipulate that now. This is kind of messy. The technology is still being worked out. What you can do is you can reset this pattern pharmacologically, for example. You can reset this pattern to say, no, two heads. And then when you cut, it's sure enough, it makes a two-headed animal. Now, here's the really important part. This bioelectrical map here is not a map of this two-headed animal. This bioelectrical map is a map of this one-headed guy. And what it is it's a latent memory that is in fact not used until and unless he gets injured. In fact, you might think of this as a basement of a counterfactual thought, you know, that amazing mental time travel that enables you to have memories and predict things that aren't happening right now. That's what this is. This tissue has it's a one-headed animal where if you ask what does it think a correct planarian looks like, the answer is, well, a correct planarian should have two heads. Now, it's not going to act on it. It just sits there like this until it gets injured. But once it gets injured, once we make that cut, the tissues consult this pattern. They say, oh, two heads. And then they go ahead and they make two heads. So a single planarian body can have one of two different representations of what a proper planarian is going to look like. In fact, I'm sure it can store much more than that, but we've nailed down these two. Now, so it's got a kind of a counterfactual, it's able to store a counterfactual memory of what a correct planarian is going to look like. Now, why do I keep calling it a memory? Because if we take this two-headed animal and we cut off the primary head, we cut off this the ectopic secondary head, that middle fragment in plain water, no more manipulation of any kind, with its normal genome, we never touched the genome is still wild type, will continue to make two-headed animals. So the reason I call it a memory is because it has the properties of a memory. It's long-term stable, meaning that this goes on forever. As far as we can tell, once you've made two-headed animals, they stay two-headed forever. It's rewritable because bioelectrically we can shift two-headed to one-headed and one-headed to two-headed, and two-headed we can shift them back. This, by the way, is a video if you're wondering what two-headed animals do in their spare time. This is what they do. It has conditional recall, as I just showed you. If you don't cut it, a one-headed worm with the wrong pattern will just stay that way. And it has discrete possible behaviors. So what we've been doing is really trying to understand, first of all, the state space of that electrical circuit, right? So that electrical circuit, we can make a model of it, and it has the specific tractors in that state space that correspond to one head, two head, and so on. And then what we want to do is merge this tightly to the current connectionist paradigm, where people have some interesting computational tools for asking how do electrical networks remember things. In particular, how do they restore so pattern completion, right? So when you've wiped out some of the information, how do you get it back? And just in general, link together the understanding of thermodynamic understanding of what happens in neural networks with the idea that these cells literally are a protocognitive agent that's trying to remember where in anatomical morphospace it belongs and can navigate there if that memory is correct. And so now I've shown you that the question of how many heads does not exist it's not hardwired into the genome. The genome just gives you some default hardware that all other things being equal settles into one, but there's other attractors, and you can reprogram that to 2 or 0. But you can go further than this, and you can actually find attractors that not just for head number, but for head shape. And that means that you can make planaria that have so here, this is a triangle Planarian head where what we do is we amputate the head, we sort of confuse the rest of the network as to where it's supposed to go bioelectrically with ion channel drugs and such. And then they land in different attractors. So sometimes you will get you can get a flat head like a P. felina, you can get a round head like an S. mediterranea, or you can get a normal triangular head. The distance here is about 100 to 150 million years. Again, remember, there's no genetic changes, nothing wrong with the genome of any of these things. They develop the brain shapes and the stem cell distributions of these other species. So in that space of possible goals that this collective intelligence can have are some regions for head number. There's some regions for head shape, and in fact, there are other regions that aren't even used by natural evolution at all. So you can make planaria that are these weird spiky forms. You can make cylindrical kind of forms like this that have a different symmetry type, or in fact combinations. So back to Darcy Thompson, we are now able to force that system to regions of the morphospace that evolution doesn't use, probably because these are not ecologically competitive. So what we're working on now is a kind of full stack model where you start with a molecular genetics of well, what channels are expressed in my cells. And so this is where the genome fits into. But now you want a tissue level model of the physiology, saying, okay, well, now that you've got these kind of electrical components, what does that electrical circuit do? What are the properties? What are the memory properties? What are the computational properties? And then work that up to a whole body level kind of set of algorithms, and eventually to human understandable algorithms that enable intervention, so that when you go in and you say, well, okay, I would like this number of heads with this particular size, this is where you want to be programming, not back here, right? And that's what we're working on. One, I'll just show you very quickly, because I want to finish off with a couple of other things. Just very quickly, another biomedically relevant example of exactly this strategy is this. This is a tadpole brain. So you see here forebrain, midbrain, and hindbrain, and we saw that one of the things that happens when you expose these animals to nicotine or alcohol or make certain mutations, their brains get all screwed up, of course. And so what we want to do is to understand how can we communicate to those embryonic cells to rebuild a correct brain, even though they've been injured in various ways. And so we made a computational model of the electrical pattern that tells this brain what the correct size and shape should be. And then we took an example, and we took both chemical injury examples and this one. This is my favorite. This is a mutation of a gene called Notch. So Notch is a really critical neurogenesis gene. Here's a normal brain. This is a Notch mutant. So you can see forebrain is gone. Midbrain and hindbrain are basically a big bubble. These animals lay on their sides. They have no behavior. They have profoundly brain damage. So what we did was we asked that computational model, what ion channels would we need to open and close to resolve this electrical pattern back to the correct state? In other words, this cellular collective does not know anymore how to navigate that morphospace. How do we show it where to go? The computational model made one very specific prediction, a certain channel called HCN2. And when we triggered that HCN2, either chemically or genetically, the animals got their brains back. They got their brain structure, their brain function, they got their IQs back, despite the fact that this really important notch is still mutated, or despite the fact that they've been exposed to nicotine and alcohol and all these nasty toxins. Their IQs are now indistinguishable from controls. So what this is telling us is that we can use these computational models to learn to communicate goal states to that the cellular collective in a biomedically relevant way. And what's amazing about this is that there are certain hardware defects. I mean, Notch mutation is a hardware defect. The protein is just it's a dominant mutation. It's wrong. Some of these hardware defects are fixable in software, because we simply expose these animals to an HCN2 opener drug for a brief amount of time, and it fixed the bioelectrical pattern because the model understood how, and we can go into detail if anybody cares. So this is the kind of biomedical strategy we're using, which is to have a computational platform where you see a particular problem, and then you ask the platform, okay, but what signals do I need to give to the cells to get back to where we need to be? And so in the last few minutes, I'm just going to go through a couple of take all of those details. Let's back out from those details, because I think it's important to have shown you that these ideas of scaling of competencies lead to predictions and lead to kind of empirical research programs. I want to pull back into a conceptual idea and start thinking about what this means. And so my framework is called TAME, or Technological Approach to Mind Everywhere. And so let's just run through this. So one thing to keep in mind is that while one thing that happens during evolution is the scaling of cognition. So individual cells have single cell level goals. What are those goals? Well, to make for every cell to make 2 cells and to go wherever life is good and metabolics are working fine. When they work together, those goals scale up massively. So now these cells are working towards this goal, the making of a limb. No individual cell knows what a finger is or how many fingers you're supposed to have, but they all work, the collective works together on this goal. How do we define goal-directed activity? With James, we say that, well, it's the idea that we know that's the goal because if we injure it, if we deviate it from that state, it will do what it can to get back to that state, and that's when it will stop. Okay, so that's what the collective does. But this can also break down. So these are human glioblastoma cells where individual cells in this collective have disconnected. When they disconnect electrically from that network, they roll right back to their ancient unicellular lifestyle. At that point, they just treat the rest of the animal as external environment. Cancer cells are no more selfish than normal cells. It's just that their self is smaller. These cells all serve this gigantic self like this. When that process, that glue, that cognitive glue breaks down, they start to serve a very small self, which is a single cell. And so you can start to make models of individual cells gathering into electrical networks, which immediately provide a bit more memory and more ability to store past information and make predictions about future information that allows them to handle larger goals. Again, biomedically, what that looks like is we put in that oncogene and then we say, okay, you've got the oncoprotein, the cells are going to try to make a tumor, but what we're going to do is bioelectrically force them to stay connected with their neighbors, and the network completely normalizes. So here, this is the same animal. So here's the oncoprotein, very strongly expressed, in fact, all over the place. There's no tumor because we managed, we force these cells to stay electrically connected to their neighbors. And of course, this is also now moving into oncology. So what I have is this idea that one way to compare and contrast very diverse intelligences, regardless of their construction, is by the scale of the goals that they can maintain. And there's a lot more to it that I don't have time to go over now. We can get into it in questions, but the idea is that what we ought to be asking about any of these things is, what are the competencies in what space, and what are the largest, in terms of space and time, the largest types of goal states that system can maintain. And of course, we are compound intelligences. And so the idea is that each of these larger scales is bending or deforming the option space for the objects underneath it, so that what the objects underneath it end up doing is falling down various gradients, which is easy for them to do, but the actual shape of the whole space is actually set by the systems above them. And so what evolution has apparently done is basically pivoted that same trick of bending the space and then learning to navigate a space across different kinds of spaces. We start out knowing only how to navigate metabolic space, eventually physiological space, eventually transcriptional space, eventually morphospace. And that became actually behavior in 3-dimensional space that we now all recognize as intelligence. And the way that we've been modeling that happening, and again, I'm not going to go through it in detail unless somebody asks, is the scaling of the ability of collectives to store larger goal states. So whereas a single cell can store a very simple goal state like a pH level or something like that, networks, when they couple this homeostatic loop, immediately begin to measure bigger states. They remember bigger set points, and they can take larger actions, which allows them to do things like this. That is the scaling of intelligence. And so once we understand this, we can think about this more broadly. And so I just want to show you at the very end a couple of new things, which is and again, this is I have to do a disclosure here. This is Fauna Systems, a company that Josh Bongard and I have for biorobotics. What we asked is a simple question. When we take cells liberated from their normal environment, what are they going to do? What's their capacity to re-envision their multicellularity, and to what kind of life will they have when we release them from their various constraints? And so what we did was we took some skin cells from an early frog embryo. We put them in a pile. Now, I'll just say ahead of time, you wouldn't know what they would do, right? If you liberate these cells, well, they may just die. They may spread out and crawl away from each other. They may form a 2-dimensional monolayer the way that cell culture does. What they instead do is overnight, they come together into this kind of structure. The flashes, by the way, are calcium. And what they make is something that we call a xenobot. Xenopus laevis is the name of the frog, but this is a biorobotics platform, so xenobot. And what you'll see is here, it makes a coherent little proto-organism. The cells have cilia, these little hairs that normally redistribute mucus on the body of the frog, but here they're using to swim, to move against the environment. They can go in circles, they can patrol back and forth. Here's some collective behavior. And these guys are interacting with each other. These ones are doing nothing. This one's taking this kind of long journey. They can do mazes. So you can see this one here. It will swim along. It's going to take this corner without bumping into the opposite wall. It doesn't need to. It knows there's a corner there. And at this point, some kind of internal dynamic that we don't know about kicks in, and it decides to turn around and go back where it came from. Okay, so there's all kinds of spontaneous. There's no water flow here. It's still water. We're not pacing this thing in any way. And they're completely autonomous. These little pieces of skin. There's no neurons. There's no brain. In fact, if you look, they have some very brain-like calcium dynamics, except that there are no neural tissues here. They can regenerate. So if you cut them in half through this little hinge, they will exert massive force through this 180-degree hinge to seal themselves up back into their shape. And the most amazing thing that we had no idea about was discovered by AI, which is Josh Bongard's lab did some computational modeling of these guys running around in an arena with little blocks strewn about. And what they noticed is that if you model the way that these xenobots move, they actually make little piles. Okay, and so they modeled all kinds of different shapes, and we noticed these. This is the kind of the tracks that they do when you leave cells around, and they make these little piles. And we thought, well, what would that actually be? So we gave them some loose cells to play with. So these are xenobots. These little white specks are just loose skin cells. And we found something amazing. We found that if you make it impossible for these creatures to reproduce in the normal froggy fashion, and so they can't do any of that, but you do provide them with skin cells, what they do is within 48 hours, they figure out how to do von Neumann's dream, which is making a machine that assembles copies of itself from material it finds in its environment. So what they do is they run around and they collect these skin cells into little piles. Now, because much like we did when we made the xenobots, they are working with an agential material, not a passive material. Then once they make these little piles, well, guess what the piles turn into? The next generation of xenobots. And then those piles mature and they run around and they collect and make new piles, and that's your next generation, and they keep going. So this capacity, as far as we know, this kinematic replication does not exist anywhere else in the biological world. And I'll just, I'm almost done here, just to say that there's something amazing about this evolutionarily. So here's the Xenopus laevis genome. We didn't engineer the genome. We didn't do anything to the genome. We engineered by subtraction. We released these skin cells from their normal constraints. So if you ask yourself, what do skin cells want to do? Normally, if you just look at normal development, you would say, well, they want to be a two-dimensional, boring, passive layer on the outside of the tadpole, keep out the bacteria. That's not actually what they want to do. That's what they want to do. That's what they do when they're bullied into it by instructive interactions by the rest of the embryonic cells. On their own, they want to have a much more exciting life. So here's a couple of paths. This is the normal Xenopus path. This is the Xenobot path. They want to create something like this, which is very active and three-dimensionally shaped. They have their own developmental sequence. This is an 80-day-old Xenobot. I have no idea what it's turning into. And then here it has some of their own behaviors. So the ideas here are that what did the frog genome learn during evolution? Yes, it learned how to be a frog in the right environment, but that's not all that it learned. It also makes a set of hardware that can actually live in multiple different ways. So what evolution seems to do is not provide specific solutions to specific problems. It makes problem-solving machines that can solve these problems anew. And in fact, so there's never been any xenobots. There's never been selection to be a good xenobot. This is all something that's figured out kind of on the fly from the extremely competent hardware that evolution has provided us with. So not going to go through all this in detail, but just to say that here are kind of the basic messages of this of my framework. And we can discuss that. And the final thing I want to say is simply this: because life is so good at making decisions on the fly, and it's so incredibly interoperable. So you can make chimeras of cells with other things, including other tissues or nanomaterials or anything else. Basically, every combination of evolved material, being DNA, cells, tissues, whatever, with any kind of a designed material and any kind of software is some possible agent. So when Darwin said endless forms most beautiful, being impressed by the variety of life, all of natural life is a tiny dot in this incredible option space of combinations and we already have some of these, so hybrids and cyborgs and various kinds of biorobotics and chimeras and things like that. But this space is massive. And so whereas in the past, you could make decisions about relationship to other agents, right? By meaning your ethical obligation to other agents, you could sort of walk up and knock on something, and if you heard a metallic clanging sound, you could conclude that, okay, this was made in a factory. It's probably pretty boring. It has very little capacity for novelty or cognition, and it's okay for me to take this thing apart into its pieces and do whatever I want with it. Versus if you were to touch it and you feel something sort of soft and furry, you would say, this is an evolved agent. I need to be nice to it, and so on. Those criteria, where did it come from and what's it made of, are not going to survive the next decade or two as any kind of a basis for understanding how to relate to these things, because we are going to be surrounded by agents that are completely alien. They have no touchstone as far as phylogeny. So trying to figure out what it is that you can expect of them and what it is that you owe them is going to be a very hard and very important task that's going to make kind of Darwin's revolution seem like child's play. So that's it. I'm gonna end here by saying that if anybody's interested in the details, there are lots of papers that dig into this stuff. I want to thank all of these collective intelligences, which are the postdocs and the students that did all this work. Lots of collaborators are funders, to whom I'm very grateful. Illustrations, most of what you saw was made by Jeremy. Again, our disclosures here, and of course the model systems, which as always do all the heavy lifting. So I will stop there and take questions.
Thanks, Michael. So there's not that many of us, so feel free to ask questions directly, everyone. And if you want to, you can raise a hand and we will get to you eventually, but we can leave it free form. Yes, Sahil?
I was just applauding.
Okay, sorry. So first question.
May I?
Yeah.
So it looks like the electric map is something that emerges from the behavior of the cells, but then kind of affects them back. That the intelligent part is this creation of the feedback loop, and I think that there is some resemblance to consciousness where we affect our behavior depending on how we define it, but the consciousness is definitely generated by the action of the cells in the brain or in the body overall. Do you see any connection with that?
Yeah, I can say two things about that. One is that you're right in that I don't know of anything. So if you look at all the theories of consciousness in terms of what might be the necessary and sufficient properties, and why do brains sort of have this property and so on, none of that all of that exists outside the body. So in other words, every criterion, I've never come across a criterion that cannot be found in these other kinds of constructs. So yeah, I think there are very strong parallels here, both in terms of this kind of large scale control, bidirectional control loop that you're talking about, and just the basic properties of electrical signaling connections, whatever else that you think is driving the property or the capacity for consciousness. It's we should definitely be open to it in all of these contexts that I showed you. It's not going to be verbal, so you won't be asking it to report on what it feels like, but you can't say that it's not there. The other thing I will say is this: it's true that the shape of the bioelectric information is strongly modified by the hardware, right? So it's absolutely true that the cells have to express certain ion channels, otherwise the electrical patterns aren't going to be there. But there's an important phenomenon here that has to do with the free lunch, the idea of free lunches in mathematics and physics, which is this. I'll give you a simple example. If your evolution and you evolve a particular ion channel that is voltage sensitive, so now you're a voltage sensitive current conductance, so you're basically a transistor, right? So you evolve this physical thing that's a transistor. A very simple combination of a few of those gives you a logic function. So you can make ands and ors and things like that. Once you've done that, you have a truth table that you're really computing, right? This truth table. And in fact, if you can do not and, then you have a really nice capacity. You can do a lot. You didn't have to evolve any of that. You didn't have to evolve the truth table. All you evolved was a voltage gated current conductance. Now, where did that truth table come from? There's a much simpler version in terms of geometry. If you're evolving a triangle and you get the first two, you evolve the first two angles, and that's tough. You got to find the angles. But after you evolve the first two angles, you don't need to evolve the third one. You know what the third one is going to be for free. And that's an amazing thing. It's not quite a free lunch, but it's a very cheap lunch in the sense that you get more out than you put in. In other words, by evolving these specific interfaces to aspects of physics, mathematics, computation, the physical machine is an interface to more than you put in an important way. So much like our bioelectrical simple states are in kind of an index into a set of organs that you might build. All of these things are an index into, I don't know what, some sort of Platonic space where these things like the truths of geometry, the Archimedean machines, all the computational laws and everything else, the distribution of primes, all these things hang out, and you don't need to evolve those details. You just need to evolve something that can manifest all of that stuff. So all of that is a long detour to say that when evolution discovers certain kinds of ion channels, the circuits made from those ion channels have amazing properties. They self-organize, they do symmetry breaking, they make self-healing patterns, they do pattern completion, they rescale, they have memory, meaning that if you change them, they hold on to that. They have all these amazing patterns. It's interesting to think about where that came from. Evolution did not specifically evolve all of that stuff. All that evolved was the physical implementation, but all that other stuff you get basically for free. So I think we have to strongly think about this idea that when you ask where do these, the Xenobots make it very plain. Like you ask, okay, where do the behaviors of and the shape of the Xenobots come from? Given that selection is not the right answer because there's never been selection to be a good Xenobot. Right. And so where did that come from? So we have to now understand that these things come from genetics sometimes when there's been a specific evolutionary pressure for it, but also generic laws. And it's the kind of stuff Stu Kaufman has been talking about these network properties and so on, which is massive. And I have a feeling in the end is probably going to end up being the vast majority of what we're interested in.
Just a very quick follow-up, because you triggered another thought. With the Xenobots, is it possible that a long time ago in evolutionary history, there was a behavior of cells that was useful, and it just wasn't manifested in the multicellular organism? But when you took out those skin cells and released them, it kind of reactivated a pattern of behavior that was there all along.
I mean, it's true in that I think it's absolutely true that what was there is the ability to form a coherent organism in various ways. That I think is fundamentally what all genomes learn. What evolution learns is how to make hardware that can, under different circumstances with different competencies, can still get various jobs done. And that is something that evolved a very long time ago. And that's used in normal embryos, although we don't really pay attention because they always make the same thing. And it's also used in these chimera constructs. But the more specific claim is that that would be that, okay, in some point back before behind the vertebrate lineage, there was some organism that lived exactly like this. And that's what this is. This is basically an atavism back to some ancient stage that it had. It's very unlikely in this case, but it's going to be completely impossible for some of the new things that we're building that are just so unbelievably alien that there's just never been anything like it. And so it's going to be all of these things are going to be a combination of some capacity, some computational capacities that cells learned basically from the time they were bacterial biofilms, up and a few things that are actually repurposed, like, ciliary motion is good for moving mucus, but it's also good for running around if you're small. So
thank you.
Thanks.
Sahil?
Yeah, hi. Thanks for the brilliant talk, Michael.
Thank you. Thank you.
I loved how it ranged from concrete to abstract and philosophy to experiments.
Thanks.
So when you're running these experiments and you have some model of what their goal is, and what I'm understanding is that you're sort of providing specifications, large-scale specifications, and they figure out the implementation details on their own. Like you said, you don't need to micromanage. You will induce an entire eye with your bioelectric networks. How often does it happen, or how hard is it to manage perturbing a goal versus providing a specification? Does it often happen that you accidentally disrupt the goal?
Yeah, great question. So I'll say the specific thing, and then there's a more general kind of concept here. One of the nice things that this interface does, and I think this bioelectric interface is the subject of evolutionary pressure in both directions. On the one hand, it needs to be simple enough that it's easy for evolution to modify it, so that evolution is basically behavior shaping of these cells, right? You got cells with baseline behaviors, and if you want them to be a two-dimensional skin layer, you have to give them specific signals. So all of the biochemical, biomechanical, and bioelectrical interfaces have to be simple enough such that the search space is not horrible and rugged, but it's easy enough for other cells to sort of manipulate each other to do what they need to do. That's important for evolvability. On the other hand, you don't want them to be too simple, otherwise you get hijacked by parasites that are going to manipulate. In fact, we've shown examples where bacteria jack into this bioelectrical system and screw up the patterning of the planaria, for example. So, but the overriding property here is that this bioelectrical interface is in fact very forgiving. It's akin to, if I can use a really stupid example, when you're typing on your computer, it doesn't, within limits, it doesn't matter how hard you hit the keys, right? You might hit it pretty hard, you might hit it very softly, it doesn't matter. It's a digital interface. And so the bioelectrical pattern is kind of like that. It's very robust to small differences. In fact, when we started doing this 20 years ago, we had very little control over what we were doing, and we noticed that that's okay. The interface kind of takes care of that. But absolutely, if it's possible to interface with it at every level. So I can do things that turn a single cell into the wrong cell type, for example, and that's kind of very counterproductive. But I can also do things that trigger a specific organ. In fact, you can even go higher level than that. In our frog model, we show that the exact same treatment that triggers tail regeneration also triggers leg regeneration, meaning that that particular signal doesn't say what to build even. It says build whatever normally goes here. So that if I apply it to a leg wound, the cells know that, well, if we're going to regenerate, we're making a leg. And if I do it to a tail wound, that's what they make. So there are different levels of interface. And I think this is teaching us something much more general. One of the things I have in my framework is this spectrum of what I call a spectrum of persuadability. If you sort of imagine it, on the left side, you have things that are very simple physical machines, like a mechanical clock. So the thing about those kind of machines is if you want to control them and if you want to change what they do, you have to rewire them physically. You can't convince them of anything. You can't train them into anything. You can't argue with them. You just have to physically rewire them. You sort of move forward, and then you've got a machine that's like a thermostat. Well, now that's a bigger, that you've got some more options there, because there, if you know that it's a thermostat and you can change the set point, you don't need to rewire it. You don't, in fact, even need to understand how the rest of it works. You just need to know functionally how to change that set point. Then you can move further, and you might find something like an animal which you can train with rewards and punishments. And there you can do even better, because this is why humans have been able to train animals for thousands of years and knowing zero neuroscience at all. You don't need to know what the encoding is. You don't need to know where the set points are. You don't need to know how anything works between their ears. What you need to know is what is the currency of reward and punishment, and then the fact that they are in a learning system. And then you can sort of go further beyond that, and you get linguistic kind of reasoners like humans, where you don't even need to provide rewards and punishments. You can provide cogent reasons to do things, and then they'll do things. So what happens during that continuum, and I've only laid out like a couple of steps on that continuum, it's of course very dense. What happens is, as you go to the rightward towards greater competencies, the amount of detail you need to know goes down. The amount of complex work that can happen from a very small input goes up. So efficiency is massive. And you lose control over the microstates, and you gain much better control over the macrostates. And so for me, all agency claims are engineering claims. So when you say to me that this system, I think this system has zero agency, meaning that it's not cognitive at all, or I think this system is at the level of a thermostat or a better thermostat that resists you changing the set point or something like that. What you're really telling me is, if I were to try to interact with the system in an engineering context, meaning use it to build something or control it in some way, this is the set of behaviors I can depend on. This is the set of competencies I can, I can depend on it to, without me having to micromanage it, to do X, Y, Z. So to me, all of these kind of claims are engineering claims. And for the example of morphogenesis, there are different levels that you can take advantage of across that whole continuum.
Thanks so much. I do have a follow-up question, but I think we're very much out of time, so I'll let Anna decide that.
I think you can go ahead.
Right, so it's still unformed, but when I'm thinking about your persuadability spectrum, do you find, I'm trying to connect this to the collective intelligence thing of how that operates. So do we find cells of like equal levels of hierarchy sort of using this these complex interfaces at various levels with each other? So if I imagine one organism with many interface levels, and then another peer with many interface levels, do they have complex levels of interaction? I wish you could see my hands so I could paint a picture, but I hope that's clear.
Yeah, I'm not sure if I've got the whole question, but one thing I can do is I'll go back to sharing the screen just for a moment. I want to show you one example of hold on, this isn't the slide. Hang on, just one second. Skip slide. Yeah, here we go. Yeah, so I want to show you a quick example of how the scaling happens, and maybe that'll help. The thing that scales up with cells, and it's not just bioelectrics, it's a number of pathways. The thing that scales up is two things. There's a kind of mind meld, which I'll show you momentarily, and then there's the scaling of stress. So one of the important things about becoming a larger scale intelligence is that the local stresses of individual subunits, the way I define stress is that error function, that delta between your homeostatic set point and wherever you are now. So that thing that drives you to keep going through that loop to reduce that error. You have to start enlarging that stress by sharing it with your neighbors. And you can sort of think about, if you tell me what you're stressed out about, I can immediately tell your level of cognitive sophistication. So if you're stressed out about the local level of glucose and that's it, you're probably bacterium. But if you're stressed about the financial markets over the next 100 years, you're probably a human, and then everywhere in between you can put something else. So here's how you scale cognition with cells. Here's a typical scenario. Here are two cells. This cell secretes some kind of signaling molecule, and it swims over and it hits a receptor on this cell. This cell, it's very easy for this cell to determine that this signal came from the outside. And so the boundary between them, the boundary of the self between them is very strong, because it's very clear to the cell that it came from the outside. It can ignore it. It can whatever. It can do whatever it wants. Now imagine what happens when these two cells are connected by gap junctions. Gap junctions are magic in the following way. because they connect directly internally to the milieu of the two cells. Let's say that this cell was there was some event that happened. Let's say it got poked or something happened, and there's a calcium flux. And now here's this calcium flux, and this small signaling molecule diffuses over into this gap junctionally connected cell. Whereas before, this cell could easily tell that, hey, that's not my information, that's my neighbor's information, and I may or may not do anything about it. This cell now has that same information internally. These gap junctions do not preserve ownership. In fact, they wipe ownership of memory information. So when this calcium gets here, this cell, which also uses calcium for exactly the same memory, cannot tell that this is now a trace of somebody else's experience. Okay, so it basically wipes that strong identity distinction between the two cells. They share all of the key memories. Once you share your key memories with someone, it's like a mind meld. It becomes very difficult to keep individual identities, because now something that happens over here is felt over here by this cell, and this goes, of course, in a bigger network that just scales up and up. So it erases the individual identities and enables this larger unit to have more computational capacity, but physically it cannot avoid now being able to respond to things that happen somewhere else. So the memories that now it has, it's a false memory as far as this cell is concerned, but it's absolutely a veridical memory as far as the new individual, the new collective individual is concerned. And so that memory wiping property helps to build larger individuals. And then of course, the other thing it can't do is it can't defect. So if you're this cell and you want to do something nasty to your neighbor, you can't, because whatever you do immediately and directly comes back to you. It's just impossible to defect. So that forces cooperation. So that kind of process, and we still know very little about it. I mean, we're doing a lot of work on this, but that scaling process of connecting into these electrical networks with these very special elements that are direct connections tends to scale the individual, and then sometimes that can fall apart too. But that's where the scaling comes from. Does that help at all?
Yeah, I really think it does. Can I have a quick note about how the gap junction comes about? Did you say that was just this type of cell, or like what is the difference between the top and the bottom picture? That wasn't fully clear.
Yeah, well, simply that gap junction is something that was discovered by evolution at the beginning of the Metazoa, and it's a protein. It's a collection of proteins that make these little barrels that they're like submarine docking hatches, where they basically just, this cell expresses a certain kind of connexin protein. There's 6 of them. They sit in the membrane. The other cell does the same thing. They kind of go together like this, and that's it. Now they've got complex gating, so cells can open and close them. There's all kinds of complexities here, but it's a trick that evolution learned to make networks. That's what it does. It basically makes, when you look at models of artificial neural networks and those edges, right, that are between the nodes, that's what this is. These are basically those edges between the nodes, and the whole thing ends up trainable and larger goals and all that stuff.
Yeah, thank you so much.
Kai, I think you are next.
Sure. Thanks for the great talk. My question is, if you could say a few more words about how you kind of identify the change that is needed in order to have a certain effect. So I'm thinking of the example of the tadpole with the kind of notch mutation, where you figured out like what to induce. If you could just say a few words about that. Great.
Sure. Let me see if I've got all the slides here. I may not, but I'll just tell you the story. Okay, so here's what we noticed. Let's just look down here. So this is a rough voltage map of the early ectodermal tissues that are going to become brain. And one of the key things about this is that you can see a really tight voltage difference on the edges between these cells and these cells, which this is where that voltage difference is what tells these cells that's where the edge of the brain is going to be. Okay, the difference in voltage is what demarcates the boundaries around specific organs, right? So when we looked, we noticed that one thing that happens to all of these embryos when they get affected by toxins, by mutations, by many different things, what happens is that the edges get fuzzed out, meaning that the edges become really fuzzy. And you can sort of see that here. This is some quantitative data where the correct, if you just drop a scan line like this across the middle, what you see is that, you know, the voltage low, 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 high, low, 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 like that. And it's literally that difference between here and here that is necessary for the brain. So if everybody goes up here, it's terrible. If everybody goes down here, it's also terrible. It's literally this pattern that you need. So all of these defects come about by a flattening of this kind of in either direction, doesn't matter which direction, of this kind of thing. So we asked the model a simple question: if the gradient here becomes fuzzy, and so the difference is very small, how would we enhance that? So what you're really looking for is a contrast enhancer. If you're in Photoshop, that's basically a sharpen filter. If you work in the retina, that's basically edge detection. What you're looking for is to make that gradient much sharper. And the computational model found for us an amazing ion channel called HCN2 that has the following property. If your voltage is low, it leaves it low, but if it's even a little bit high, it cranks it up. So that basically, again, contrast enhancement, that anything that's sort of, you know, that kind of gentle low hill that codes for a bad brain becomes yanked upwards. The low cells stay low, the kind of slightly higher cells get jacked up, and in the end you get this pattern back, and that pattern is what's required for these cells to respond to where the edge of the brain should be. So what we found is the strategy that we're exploiting here is sharpening the voltage gradients between organ boundaries. Now, that's not going to, I guess, probably that's not going to be suitable for everything. There are going to be other reasons this goes wrong, and we'll have to come up with other strategies. But for a wide range of things, that works. And in fact, the wild thing is that we developed this strategy with very specific voltage data on the early brain, because you can see that the nice thing about the brain is right at the top of the tadpole, you can see it. So we have all these voltage data. We developed the strategy. 2 weeks ago, we published a paper showing that you can use the exact same strategy on the gut and the heart, where we have no idea what the voltage is in those organs because you can't see them. They're deep inside the embryo. We don't have any technology to see that, but that's okay. The exact same technique worked. So I think, as usual with these things, you can get out more than you put in, meaning that this is going to be workable across a range of applications. But there will be other things where the edges are not the problem. Some other thing is the problem, and then we'll have to go back to the model and invert the model again and ask how you solve whatever the other problem is.
Thanks. So to summarize, it's something, so you know, for the reasons that you explained, that a lot of the signaling occurs via like the bioelectrical interface, and the voltage is really important. And then you'll kind of compare the healthy versus deficient organisms and see if there's something you can do to kind of maneuver the defective voltage patterns back to the healthy one. Is that, like, roughly speaking,
that's what we're doing for now. To be honest, I have a feeling that this strategy is we're using this because we still underestimate the intelligence of the system. I have a feeling, but I can't prove it yet, but we're investigating it, that there's actually even more competencies here than we suspected. And I hope that down the line, we will be able to dispense with this actually and produce signals that are much higher level, so that instead of saying, now remember, your brain boundaries have to be exactly here, right? That's how we're doing it now, which is better. That's a lot better than saying, oh my God, this cell has to be this kind of neuron, the next cell has to be this kind of neuron. Like, that's pretty hopeless. We're in a better place where we can say, just this is how big your brain is. Now go. That's better. But I have a feeling that eventually, when we really understand the competencies of this system, we're going to be able to do even better than that. And we're going to be able to do what we did in the leg and just say, just build what normally goes here. Trigger again your memory of a correct brain and rebuild that, and that's it. Like, I want, as a worker in regenerative medicine, I'm lazy. I want the least amount of effort, the least specification that I want. And I have a feeling that even this is too much. I think we haven't discovered the real competencies of it yet. But that's a hypothesis. That's a hypothesis. 10 years from now, we may find that, okay, that's it. That's where this system flattens out. There is no more competency, and we have to do it like this. It's okay. But I bet there's a higher one.
All right. Thank you so much. That's really cool.
Thanks.
Ash, do you want to go next?
Yeah. Hi. Thanks for the lecture.
Thank you.
I would be curious. What is your favorite or promising model or framework of like abstract framework of computational intelligence or theory of individuality, where I mean, I see that you are modeling how part of the systems can be intelligent, but then they somehow cooperate to create larger complexes. And so, do you have any higher level or abstract theory behind it? I'm thinking about something like, for instance, predictive processing, or like this level of framework.
Yeah. I think, I like predictive processing a lot. I do work with Carl on some things. I think that's really important. I think there's I think the fundamental thing here, which drives everything else, and I'm going to show you, I'll go over a couple of the specifics. But I think the fundamental thing that we start out with is that biologicals and other things that may not be biological, but we don't know, that what biologicals do is they don't take past experience too seriously. Now, I know this is a crazy thing to say for a biologist, given evolution and everything else, but I think we need to think about the fact that the way that, we have this, in robotics, we have the sim to real problem, right? Where you simulate and you simulate and you simulate, and then you make something, and then the simulation is never quite good enough. If you depend on what you did in simulation, it's always often like wrong and doesn't work quite right. And I think evolution, the way that evolution has worked is that it doesn't take prior experience too seriously, meaning it doesn't overtrain. When you have a genome, it isn't perfectly, at least in most animals. There may be exceptions. C. elegans or something may be an exception, but many animals, the genome isn't overfit on prior experiences, and now it knows how to do exactly that. It's a problem-solving machine. What does that machine have to do? As soon as the juice is turned on, so to speak, once it's going, it has to figure out what are the causal control knobs in my environment. Meaning that what are the things that, with my limited metabolic and time resources, what are the things, what are the knobs that make sense turning? In other words, by sending signals and so on, which means that it has to do things like causally coarse grain its environment. It has to adopt a kind of a predictive processing framework, in that it has to start to model itself as an actor, as doing things. As opposed to a kind of a Laplace's demon, sort of infinity of causes and effects. I mean, you'll be eaten in no time if you have that view of the world. So you have to immediately start making models of what's going on. You have to coarse grain. You have to find your own boundaries. So it is not obvious. Every cell is some other cell's environment, and so it is not obvious where the edge of you as a self, where your edges are. Where do you end? Where does the environment begin? And I think that how you see that, both from us as external observers and the system itself, the boundary of the self is the smallest portion of a complex system that is required to pursue specific goals in specific environments. So that whole thing is self-constructed from the beginning. You don't know exactly where your boundaries are. Like in 2006, Josh Bongard made these amazing robots that basically they had no built-in model of what their shape was. So they just had to take some actions. They had to see what happened, and based on that, they would make a model about their a conjecture about their own shape, test that conjecture, and eventually get really good, so they could start to walk and do things, basically like babies do. So that's that was a great start at it. The next step is going to be biologicals don't even know. Never mind not knowing what your shape is. You don't know what you have. I've showed you examples. You don't know how big your cells are. You don't know how many cells you are. You don't know if your genome is correct, or if you have too few or too many genes. You don't know if you've been injured. You don't know if, all of these things. You don't know any of these things up front. So something like, active inference and a couple of other things that we've been working on has to start from scratch, figuring out where are my borders, what do I have, what are my sensors, what are my effectors, how am I going to coarse grain the world, what are the things that I need to pay attention to, and your competency at doing all of those things is related tightly to your adaptive success. And the scaling, happens. I mean, I showed you a couple of mechanisms. Like one thing that happens is this. The beauty of this of this kind of structure for evolution is that the larger scale, the larger levels make it easy for the lower levels to do things, because all they have to do really is fall down their concentration gradient, but you've bent the space for them. but the competencies of the lower levels make it much easier for the higher levels to get the job done. So for example, all I need to do is say, make an eye, and the cells know how to make an eye. And so they make the eye. I don't have to micromanage that. And by the way, if that eye ends up in some weird location, if it's in the head, it'll move to where it needs to be. And so it'll take care of itself. If it's in the tail, that's okay too. The optic nerve will find the spinal cord and then it'll be able to see. That's fine too. So all of those things, that competency has massive implications for evolvability. And it's all because baked in right from the beginning is the fact that as a living thing, you can't trust anything in advance. You don't know anything in advance. You don't know what you are. You don't know what your pieces are. You have to build, you have to construct all those things from scratch. I think Carl's framework is a great piece of that. We need out some other stuff, but that's an important piece of that. So that's what I think happens. I think evolution latched on very early to this idea that you're a multi-scale system. You don't know where any of your borders are, and you need to figure it out from scratch. And that's why we see this amazing plasticity in life.
Mm-hmm. Thanks. I'm a bit surprised that you said that the evolution doesn't, or systems don't learn that much from the past experiences, because another way to interpret what you said would be that evolution or learning learns the right variance that you need to take into account given your capacity, like information transfer between generations. But I guess that's probably just like a different framing of the same thing. So one short follow-up question. Yeah, sorry.
Well, I was just going to say, let's be clear. I mean, I'm purposely emphasizing novel aspects of the view just to sort of, pull us in that direction. Of course, I don't mean that living things don't learn through evolution from specific selection pressures. I mean, of course they do. My point is simply that both of these things are important, and that it doesn't overtrain. Right, it learns from past experience, but it doesn't overtrain in the sense of, okay, I've learned exactly how to be a frog embryo in this frog environment, and that is all that I ever know how to do. Some animals may, like I said, nematodes maybe, because they're very like cookie cutter kinds of things. But I think there's amazing generalization, and that means you don't overtrain.
Yeah.
May I have a very quick follow up? Maybe a part of. Just a comment on the last sentence, that it could be survivor bias, that the species that have gone extinct, they weren't able to adapt, so they probably overtrained.
Entirely possible. In fact, I think our early attempts at synthetic life are going to be exactly that. So people have asked me before, are there examples of life that doesn't do any of the things that I'm talking about? And now I don't think there are any, but at the beginning there might have been, and certainly we're going to be making some. Our early attempts at this I'm sure are going to be these very simple things that don't have these cool abilities. Yeah, no, I think you're right.
Yeah, I also agree with what you said. I mostly meant this. It seems to me like evolution has learned that to have a very high prior on variance, or like prevent overtraining, even when it would be locally efficient. One last quick question. So is there any reading that you would recommend that's sort of like in between, for instance, predictive processing and that connects the abstract framework with what you've been talking today?
Yeah.
If you can like point to maybe one or two papers.
I can drop me an email.
Okay,
Send me an email, and I'll send you a bunch of reading on it.
Thanks.
DJ?
Hey, yeah, so I think like some of your slides talked about like the multiple levels of organization that extends beyond the level at which we consider primarily thinking about biological organisms. So things like social superorganisms, cultural evolution, and those sorts of things. And I was curious to know if you had like thoughts on like how this entire framework actually extends to like the layer of cultural evolution, or like the layer of social superorganisms, communities, tribes, or those sorts of things, anthills, colonies, etc., etc.
Yeah. So I'll say right up front, I have zero expertise at that level, right? So I'm not a social, psychologist or anything like that. So I don't know. But here's what I feel strongly about, that all of the important things that we've talked about today are scale free, in the sense that this isn't really about gap junctions. It's about a certain type of relationship between your subunits. And it's not really about ion channels or bioelectricity. It's about the way that networks process information. So all of these things are scale free in an important way, which means that we have to let go of our idea that, okay, an agent has to be of a certain size and a certain duration, and that things that are very small or very big or very distributed or very weird can't be agents. I think we have to let go of all that. So for example, thinking about all of evolution, a whole lineage, let's say, some millions of years of alligators or something like that, that whole lineage, thinking about that as one agent where every individual animal and its genome are a hypothesis of that agent about the environment, right, to be refuted or not, those kinds of things, or looking at, incredibly large. I mean, at one point I was talking to some friends about designing a gravitational synapse, basically, right? Because a planetary scale, solar system scale synapse, where basically can we design a system of physical bodies in space where certain things are happening, and then passages of masses through that system can basically be remembered and read out later on, right? So if that's true, if you can build things like that, you can make galactic scale neural networks and so on. So I think all of those are control knobs that we can twist to our heart's desire. And so from that perspective, large scale organizations may well possibly have some of the properties that we're interested in, but that's an empirical question. So if you make a model of a power grid or a political structure or a water distribution or an economic whatever, you can ask questions about it. Does it learn? Does it respond to stimuli in a particular way? We did that with a couple of years ago with gene regulatory networks. We took models of gene regulatory networks, which look very dumb and very mechanical, but we found out that actually they have 6 different learning capacities, including associative learning. And you wouldn't know that if you just assume that, well, it's a piece of physics, it doesn't learn. So you have to do empirical experiments. And it's entirely possible that some of these larger scale structures have the kinds of capacities that we talk about. We have to find out about them. And that actually leads to a very important question, which is, can we could a lower level subunit gather evidence to suggest that it was in fact part of a larger intelligence? Like, how would you know? So if you were a neuron in a big network and you were smart enough to do so, could you ask yourself, gee, is my universe sort of cold and mechanical and doesn't care what I do? Or do I live in some kind of a mindful universe that rewards and punishes specific behaviors? And we're actually part of a grand design. Me and my buddies here in this local, neighborhood, we're all part of a grand design. And if so, what is it, right? And I assume that Gödel's theorem and something like that makes it impossible for us to really know what the design is of a larger system in which you're embedded. But maybe it's possible maybe there's some kind of math that will let you gather evidence for or against that idea that you're actually part of a larger scale cognitive structure with its own goals in some other space you can't even conceive of. So, maybe.
Right. So yeah, I think like the reason why this question, it seems interesting to me, is because like when something is a subunit, so there is a question of like knowledge of like, can the subagent even gain knowledge that it is a subagent of a like superagent or not? But there's also the question of like, even if it could gain knowledge, because there's a sense in which in corporations, like people have the knowledge that they're part of like a larger superagent, but like knowledge still does not imply control always. So it's it's not always that the knobs are things that we humans or the relevant morally relevant patients could actually turn the optimization towards their own like well-being or whatever.
Sure. Yeah, absolutely. Entirely correct. I mean, if you're part of an organization, what do you know? You know that physically you're part of an organization, the way that a cell physically knows it's part of a tissue, because you can feel the presence of your neighbors. What you don't know is whether or not that organization has goals beyond the goals of the individuals, and what space those goals exist in, and what effective actions, if any, you might take. So in a particular sense in this bioelectrics, there are specific cells that actually, by making an electrical change that will propagate outwards and be amplified and whatever, there are scenarios where the local agents can in fact change the larger scale, but you have to be able to find those, right? Those are not obvious at all. I apologize. I've got about 30 seconds and I have to hop off. But anybody that wants to keep talking, by all means, email me and we can chat offline.
Yeah, you can share your email later on. Like, yeah, thanks a lot, Michael. This was great. Yeah, thanks for joining us, giving us the talk, and maybe see you later.
Yeah, thanks very
Yeah,
much,
thank you
guys.
for the email.
Fantastic, fantastic questions. I appreciate it. Yeah, please email me if anybody wants to. All right, bye.